Hello and welcome to your monthly update from the COVIDENCE UK study. My name's Adrian Martineau. I'm based at Queen Mary University of London and I'm the chief investigator on the project. So I'm just back from the 33rd European Congress on Infectious Diseases in Copenhagen. And what I thought would be interesting today, rather than presenting our own latest findings, would be to bring you up to date with an interesting study that I just saw presented on Monday evening that was published in the journal eClinical Medicine, which is one of the Lancet family of journals. The first author is Lucy Finnegan, and the results are of a clinical trial of a new medicine which potentially might have some promise, particularly in people who have got long COVID with very prominent symptoms of fatigue. So let me take you through the story. Now, I think in order to understand the findings of this study, the first thing we need to do is understand a little bit about the drug that's being investigated. So what I'm going to do is introduce you to this chap, the mitochondrion. Now, each of your cells is packed with these little organelles, as they're called, and they're very interesting little fellows. In fact, it's thought that they were once bacteria that wormed their way into our cells and became a permanent feature of our cells during evolution. But essentially what these guys do is act as power stations for our cells. So they take in calories and oxygen and they produce energy. Now, what we know is that in COVID-19 or in SARS-CoV-2 infection, the virus can actually hijack these energy factories, if you like, for its own purposes. And what that can do is basically reduce the amount of energy that the mitochondrion can make for the host cell. And this is thought to potentially underlie some of the symptoms of fatigue, which are so prominent in people with long COVID. Now, what this trial investigates is one of a family of medicine called EMMs. And the idea is that these medicines can restore the function of the mitochondrion so that it makes energy for the cell and not for the virus, thereby restoring energy and reducing fatigue for people with long COVID. Now, let me tell you a little bit about these EMMs. It stands for an endogenous metabolic modulator. And it's a family of compounds which effectively are like master regulators of metabolic processes. Now, the one investigated in this trial has got the catchy name of AXA1125, and it's comprised of 5 amino acids and a compound called N-acetylcysteine. And you may know that amino acids are the building blocks of proteins, but they've also got roles independent of their nutritional role in terms of regulating metabolism. And we know, in fact, that this drug has previously been used to treat inflammation of the liver with some success. So we know that it has got some activity in terms of regulating energy generation in the cell. So how's the study done? Well, it's a small study, just 41 adults in the UK who had what's called fatigue dominant long COVID. So these are people with proven SARS-CoV-2 infection with sustained symptoms lasting 12 weeks or longer after infection, and with fatigue being one of the main symptoms reported by the participants. And these 41 adults were randomized using a computer program into 2 arms of the study. The active arm, which contained 21 people, who each received 2 doses per day of AXA1125 at a dose of 33.9 grams for a period of 4 weeks. And those who were allocated to the control arm, 20 of them, were given a placebo twice a day for 4 weeks. Now, this medicine comes actually in an oral solution, so it's given as a liquid medicine, and the placebo or dummy medication looked and tasted exactly the same as the active medication containing AXA1125. What this meant is that we could, or I shouldn't say we, the investigators at Oxford University, they were basically, by doing this, we're able to establish what's called a double blind. In other words, both the participants who are taking part in the study and the investigators, the doctors and scientists running the study, were unaware of whether a participant was taking the active drug or the placebo during the study. And this is really important for rigorous study design, because what it means is that neither the investigators nor the participants can bias the study results. So this double-blind, placebo-controlled design is really the gold standard for working out whether or not a medicine has any efficacy or not. So at the end of those 4 weeks, the participants underwent a battery of different tests. So-called primary outcome, so that's the major outcome of interest, was what's called the skeletal muscle recovery rate. But then there were a number of secondary outcomes, which included the 6-minute walk test, which basically is a measure of how far people walk in 6 minutes. Levels of a compound called lactate, which is the same as lactic acid in the serum. And you may know that this is a product of anaerobic respiration, which is really reflects inefficient energy generation. So if there was a benefit, you'd expect the lactate to go down. And then also they completed a validated questionnaire, which measures both mental and physical fatigue. And then the statisticians took the data, and they compared the levels of these different outcomes between people who've been randomized to receive AXA 1125 versus those who've been randomized to receive placebo. So what did they find? Well, the first news is that they saw no difference in the primary outcome of skeletal muscle recovery or in the secondary outcome of 6-minute walk distance between the different arms at follow-up. However, there was a bit of interesting signal. First of all, they saw what's called a borderline significant reduction in the levels of lactate in people who had taken the active drug versus those who'd taken the placebo. And you'll see there I've quoted a p-value, 0.07, and that p-value represents the statistical probability that this event arose by chance. Now, normally we say that any p-value of less than 0.05, in other words, a chance of less than 1 in 20, we can say, or we can interpret as indicating a cause and effect relationship in a clinical trial. In other words, we can say there's less than a 1 in 20 chance that this difference between arms arose by chance alone. In other words, there's a 19 out of 20 chance or more that the difference was due to the drug. So what we're seeing here is that it didn't quite achieve that conventional level of statistical significance, but it wasn't far off. What was really interesting, though, is when the investigators looked at the outcomes for the questionnaire. And what they found was that the fatigue score was 4.3 points lower in people being randomized to the active drug versus those who've been randomized to placebo. And what you can see there is the p-value is 0.004. What that means is there's just a 4 in 1,000 chance that this difference arose by chance alone. So that points to the likelihood that this reduction in fatigue could well be due to the medication that had been administered in the study. And interestingly, this fatigue score has got 2 components, as I mentioned, the mental and the physical fatigue score. And when they were broken down into those different components, benefits of the drug were seen for both types of physical fatigue score. There were no serious side effects. So here are the data on the fatigue score. In grey, you can see the data from the people who were randomized to placebo, and in orange, the people who were randomized to receive the drug. BID is an abbreviation that means twice a day. And what you can see on the left here is the total fatigue score. You can see that there was a borderline difference between people at baseline. So this orange group represents people who were about to receive the study drug. That's why they're coloured orange, but they hadn't yet received any because it's at baseline. So there was a small difference, but this didn't achieve statistical significance. And then at follow-up, you can see this fatigue score of 21 is lower than that of 26.5 in the control group. And this statistical test was done adjusting for baseline values. So it adjusted for the fact that the people in the orange group here did have slightly lower levels to start with. So this difference is statistically significant even after adjusting for minor baseline differences. And again, This breaks it down by the physical fatigue score and mental fatigue score in the middle and right hand panels respectively. You can see that both of these were statistically significantly reduced in the intervention arm. That's the people who received the active drug compared to placebo at day 28. That's 4 weeks into the trial. As I mentioned, no serious side effects. Very important to keep an eye on that whenever one's investigating a new medication. So what were the strengths and limitations of this study? Well, let's look at the strengths first. As I mentioned, the study utilised what we call the gold standard in terms of a double-blind, placebo-controlled, randomised controlled trial, which is the most rigorous way to evaluate whether a medication works or not. I think another strength of it is that the investigators looked at a mixture of what we call physiological or objective outcomes, things like measuring the serum lactate or measuring muscle recovery time, and also looked at symptom or subjective outcomes. So these are things reported by the patient which reflected how they actually felt. And as I mentioned, the statistical analysis was rigorous and it adjusted for baseline status. Obviously, one of the major limitations here is that it's a pretty small sample size, just 41 patients. So it may be that the drug, for example, could have an effect on the primary outcome, but the sample size wasn't big enough to detect that effect. A second potential limitation is that the only symptom studied was fatigue, and we know that long COVID has got many different symptoms in different people. I think the reason they focused on fatigue was really based on the mechanism of action of the drug, which was in terms of trying to correct the cell's energy processing metabolism function. And I think a third factor just to note is that's what we call a single centre study. So it was people just studied at a single centre, which is, of course, what you'd expect in an early stage study like this. What it means is that we can't necessarily generalise results from this small group of people studied in one centre to a wider group of participants or people with long COVID living all around the UK or internationally. So what's the bottom line here? I would say that there are maybe hints of promise here. The fact that a symptom improvement was seen and that this was associated with a borderline significant reduction in serum lactate levels, which kind of fits with the positive mechanism of action of the drug, does start to provide preliminary evidence that this could be something worth investigating further. But I would caution, however, that bigger multi-centre trials would be needed before this medication could be recommended for the treatment of long COVID. So I hope that you found that update from research of others that I saw at the conference this week in Copenhagen interesting. I'm hoping to share some other research from that conference with you in webinars in the future. But before I close, as always, I'd just like to thank you for watching the webinar and also for continuing to fill our monthly questionnaires, which continue to provide us with really valuable data looking at the long-term consequences of COVID-19. So from me and from all the team here at Queen Mary, goodbye and look forward to seeing you next month.